Thank you very much for having me. It's an honor to be here, and thank you to all the organizers. I am here on behalf of Conquenni, presenting results about our molecule tweezer array. And recently we've seen dipolar interactions within this array, and I'd like to tell you all about it. So throughout my talk, I'll explain why we're interested in the molecule tweezer array, how we produce the ground state molecules, how we then control the rotational transitions, and how, by doing all of this, we're able to see the dipolar interactions. So, to begin, if you can have a system where you have interacting dipoles that are positioned in a controllable way, there's many applications that become open. Other speakers have spoken about this, but you could engineer various Hamiltonians for studying spin systems, encoding synthetic dimensions, or even modeling exotic gauge theories. And you can also use the intrinsic coherence of molecules to make potentially highly coherent qubit and qubit gates. So we're interested in this. But to make all of these schemes possible, you need to control the dipole exchange interaction. So what I'm showing now is two interacting qubits at a fixed distance apart. And essentially, they have transition dipole moments that interact and allow you to entangle the two particles. So this is the system we're trying to control. But there's a lot of things that spoil this interaction. You can have changes in the transition frequency between the two qubits. You could have variations in the separation between the qubits, and you could have somewhat lossy qubit states caused by off-resonant fields or spontaneous emission. So what we need to study the system is have enough coherence time to see the interactions, meaning that delta F and gamma are small. And you also need to precisely control the position so that the interaction rate doesn't dephase. So this is why we're using molecules for the coherence and tweezers to keep the position fixed. But molecules have other benefits. In the lowest electronic potential, the lowest vibrational level has an abundance of states, as you all know, and most of these can be used for the dipolar interaction. And these states have essentially no spontaneous emission, and so really the loss mechanism is to do with the vacuum lifetime and the scattering induced by the tweezer. So now let's build the system. This is work that's been done in Conquen's group since 2014. And what I'm showing here is our apparatus. Red and blue are the sodium and cesium tweezers. They're focused down through an objective, and they go into an evacuated glass cell, which contains dispensers for sodium and for cesium. And the first step is to load an array of sodium and cesium. So we have overlapping MOTs, and we use light-assisted collisions to somewhat randomly load a 1/16 array of cesium. I guess we can't load the array. I'd like to touch the cables. Okay. Great. All right. So the next step is to remove vacancies from the array. So we can image the sodium and the cesium and essentially turn off the tweezers that don't have atoms and shuffle everything to the left. At this point, the tweezers contain atoms that have internal and motional entropy. And so we want to deterministically control the internal and motional states. So we use optical pumping and Raman sideband cooling. This process is pretty efficient, but I'll talk about the efficiencies in a bit. So all of this together takes about 800 milliseconds. So now the way we make the molecules is by combining two atoms in one tweezer. This is not just us, there's plenty of other groups that use this method. But what you need is to have one tweezer that can only trap one of the molecules, and another tweezer to trap both of the molecules. And that's what we do here. We move the sodium into the cesium tweezer. And then we use a Feshbach resonance to at 860 Gauss to convert the 2 atoms in one tweezer into a weakly bound molecule. The weakly bound molecule lives for tens of milliseconds, and that's enough time to use stir-up to transfer to the row vibrational ground state. So let's talk about efficiencies. With optical pumping and Raman sideband cooling, there's a chance that we don't fully control the internal state of the atoms. And if they're not in stretched hyperfine states, the 2 atoms in one tweezer will undergo a hyperfine swapping collision. And so after 1 milliseconds, we'll lose the atoms. So that's one source of inefficiency. There's also the trouble of just cooling. Unfortunately, the Feshbach resonance only works if the relative motional energy between the 2 atoms is 0. And that's why we only have 40% Feshbach production. And lastly, for stir-up, the intermediate state we use is somewhat broad, 20 megahertz wide. And so there's a one-way associated efficiency. So at the end of the day, when we form our ground state array of NaCs, for each site, we roughly have a 20% chance of having an NaCs molecule, and then these other chances of having these other cases. So if we're trying to see the dipolar interaction, and you want to have 2 molecules side by side, if you have a 20% chance of having a NaCs in one of them, that means you've got a 4% chance of seeing, small chance of seeing the interaction. So it's statistically very difficult to see. So one thing we've been working on is trying to detect cesium at high field. We've done imaging of cesium at low field, but when you're at 860 Gauss, all the transitions are shifted by several gigahertz. So we need a new laser, a new closed transition. So our scheme is basically to populate the cesium in a stretched hyperfine state of 6s1/2, and then optically cycle with a stretched hyperfine state of 6p3/2. And by doing this, we're able to get this high field imaging to work. I also want to point out that we have a new beam path that we set up for this. It uses very small beams. It's a retroreflected 1 millimeter waist beam. And by doing this, we really minimize the scattering that comes off the glass cell. And so our camera, which is detecting fluorescence from the cesium, doesn't see any background light from these beams. And so we're able to see a cesium that was there at low field with 99% probability at high field. And this imaging is quite fast. It takes about 5 milliseconds, and you can see from this curve on the top right that it's got a characteristic red detuned preference due to Doppler cooling. So now that we have high field imaging working, we want to basically detect sites that have cesium, turn them off, and shuffle the molecules in our array to the left. There's one complication though, which is that some sites have a sodium and a cesium. And as we're optically cycling the cesium, the sodium is going to have hyperfine collisions with it. So we need to somehow remove the sodium from the tweezer. So the way we do this is we're just lucky in a way. Our low field sodium light happens to be resonant with a high field sodium transition. So we blast away the sodium, we end up with just cesium in the array, and we're able to rearrange molecules to the left. And so this greatly improves the density of our array. So now I'll talk about how we detect the molecules. There's no closed imaging of NaCs that we know of. That's good enough. But so what we do is we essentially adiabatically turn the ground state molecule into 2 atoms and detect the survival of the atoms. And this lets us know that we had a molecule initially in n = 0. But we can do this at high field. And so what we have shown just this summer is that we can dissociate the molecule into 2 atoms in one tweezer, do cesium high field imaging, then go back and take all of the n = 1 population, put it to n = 0, and then re-image that. And so every experimental cycle, we're able to detect both the n = 0 and n = 1 population. So again, this is a statistical improvement, but it means that in our experiment, we're able to now collect twice as much information per cycle as we used to before. And this process, it takes 10 milliseconds per image, so it's scalable. Okay, so now I want to tell you about controlling rotational transitions. When we do stir-up, we populate NaCs in a particular hyperfine state, one where the sodium spin is 3/2 and the cesium spin is 5/2. But since we're at such a large magnetic field, there's very little coupling. The nuclear spins are not coupled to each other, and they're not coupled to the rotational degree of freedom. So essentially, even though we have 32 hyperfine states per rotational sublevel, we're only interacting with a 4-level system because we can't, it's difficult to change the nuclear degrees of freedom. So what we're trying to do now is essentially stabilize the transition frequency between the n=0 state and the 3 n=1 states. And this transition frequency is sensitive to various changes in external fields. Most notably, any intensity variation of the tweezers could cause our rotational transition to decohere within a millisecond or so. There's other effects. We have background electric fields in our glass cell due to stray charges, and those would decohere our system within 100 milliseconds. But first things first, if we want to improve our coherence time, the main thing is to basically tackle these intensity fluctuations. So to do that, we're going to try to change the polarizability of the molecule's rotational states. What I'm showing you now is the polarizability matrix for NaCs in the molecule frame. The idea is that the molecule will have a different trap depth for light that is parallel to the internuclear axis. That'll be deeper, and it'll be more shallowly trapped if the light is perpendicular to the internuclear axis. And you can see that this polarizability is decomposable into a scalar and tensor part. Because of this tensor component, the rotational states become mixed due to the polarization of the tweezer and intensity of the tweezer. And so the new eigenstates look like this, and they kind of follow the polarization ellipse. So what I'm showing you here is a elliptically polarized tweezer, and you can see that there's an eigenstate along the major axis of the tweezer, an eigenstate along the minor axis, and along the k-vector. And the eigenstates that are along the major axis are the ones that are most deeply trapped. The idea there is that that orange p orbital that you see here, it will see more light along the internuclear axis of the molecule. And so that'll have a larger tensor polarizability. So to summarize, because these p orbitals have tensor polarizabilities, whereas the n=0 state does not have tensor polarizabilities, we see this change in the, we see AC stark shifts of this transition. So Simon Cornish has this magic wavelength technique, which changes the tweezer wavelength to remove the tensor polarizability. Here, we have a different technique, which is to change the tweezer polarization. By applying 2/3 of the light along the minor axis direction and 1/3 of the power along the major axis direction, we're able to cancel the stark shift from this term. And so we're able to basically have a particular p orbital that has the same stark shift as the n=0 state. So what I'm showing you here in this left scan is that as you change the trap depth, the transition frequency from n=0 to n=1 doesn't change. And so how do we actually implement this polarization? What we have in our apparatus is gland tailors to clean up the polarization of the sodium and cesium tweezers. We then use a rotation mount with a wave plate to quickly change the ellipticity to 35 degrees, which is the magic condition. This is a very nice rotation mount. I recommend it. And with it, we're able to see this sort of intersection on the left plot. So what we're doing is we're measuring the transition frequency to that middle magic state, and seeing a point at which the transition frequency at a particular ellipticity of the polarization, the transition frequency does not depend on intensity. And we also see that our spin-echo coherence time is maximized at this angle of the wave plate. So this is all evidence that we found a way to basically make this rotational transition more coherent. We can also measure the coherence time with other dynamical decoupling techniques, both with spin-echo and repetitions of XY8. And we see that spin-echo is limited to 62 milliseconds and XY8 to 200 or so. So I think the message here is that we've found the maximum of the spin-echo coherence time as a function of angle. So we've removed the first order AC Stark shift. But what remains is electric field fluctuations, most likely in our experiment, which limits our coherence time to the hundreds of milliseconds. But that's enough to see the dipolar interactions. So just to give some more context, there's other experiments that see these dipole exchange interactions. They've been done in lattices where you have not individual control of the particles, and also more recently in lattices where you can see individual particles, and in tweezer arrays just like this one with CAF, where dipolar interactions have been seen. What makes NaCs, which is the molecule here, somewhat interesting is that it has a very large dipole moment. It's on the order of 5 dBi, whereas CAF's is much smaller. And so the dipolar interaction rate is 20 times smaller. So there's also another interesting property here, which is that the dipolar interaction is anisotropic. The interaction rate depends on the orientation of the dipoles to each other. And when you start messing with the tweezer polarization, what's interesting is that it's the orientation of these tweezer ellipses relative to each other that controls the dipolar interaction rate. So, for instance, if you're able to have your axis between your two particles and align your polarization ellipse so that the minor axis is along the axis between the particles, you're able to maximize the interaction rate. So what this means is that we're trying to basically fully parametrize the polarization of our tweezers. We want them to have the right ellipticity so that we have this good coherence. And we also want the orientation of the polarization ellipse to be chosen correctly so that we have maximum interaction strengths. The way we do this is by choosing a somewhat wacky waveplate to go into this rotation mount. We use a 0.304 retardance waveplate. And what this does is that by rotating it 45 degrees, we're able to pretty much establish the situation and see very fast dipolar interactions. So the scheme to see the interactions is shown here. Essentially, you take the two molecules in n equals 0, and you project them both into their Bloch spheres. And then the population as it evolves around the Bloch sphere, the dipolar interaction will cause entanglement to be generated. And so, as a function of time with this pulse sequence, you're going to have an oscillation in the 0,0 population and the 1,1 population. What we do with our array is we set it up into pairs. So we have 4 pairs, each experiencing the dipolar interaction. And there's something somewhat challenging about this, which is that each site has a different ellipticity. It varies by 0.06 degrees, but what it means is that the transition frequency varies site to site on the order of hundreds of kilohertz, hundreds of hertz. And this is the sort of effect we were talking about earlier that limits the coherence of the dipolar interaction. So one thing we can do is, as the particles are interacting, apply a dynamical decoupling sequence in between. And what this does is it makes the interaction robust against detunings. And so that's the idea behind having spin-echo or XY8 during the interaction. And so this is our first preliminary results in June. We see that we tried 2 different waveplates, which lets us basically control the orientation of these transition dipole moments. And we see interaction times that correspond to the angular orientation that we expect. We also see that as you bring the two particles closer, the interaction rate scales as 1/r³. More recently, we collected more data, and this is about a week old, and we're able to see this dipolar oscillation both with the Spinetto dynamical decoupling and with XY8. And we see something on the order of 4 to 6 oscillations, depending on what the interaction is composed of, depending on the decoupling sequence. And so there's several limitations here that could limit our coherence, and we're not sure yet what they are. This is pretty new stuff. It's possible, for instance, that each site has a slightly different interaction rate. And since we're averaging between 4 different pairs, by averaging between them, we're dephasing the interaction and making it effectively decay faster. But we've done simulations, and we expect to see 10 or so oscillations based on our site-to-site detunings. There's also the possibility of just having our molecules be hot. CAF, which has seen the dipolar interaction, their interaction is limited by temperature because they hadn't done Raman sideband cooling with the molecules. But we don't expect this to be the case here because everything that we've done with the molecules since Raman sideband cooling is an adiabatic process that shouldn't heat up the molecules significantly. So there's several candidates here. It's possible that we have beating between the tweezers. That could also explain it. So we're still looking into this. So to summarize, we have a new imaging technique which lets us rearrange molecules to the left of the array. And we can also use the same technique to collect multiple data points per cycle, which vastly boosts our data rate, and can scale to multiple state detection. We also have this new method of magic ellipticity, which lets us control rotational transitions quite coherently. And for now, we've seen very fast dipolar interactions with a period of which can go as fast as within 1 millisecond. So with that, I'd like to thank you and acknowledge my wonderful team, which I couldn't do this without. Yeah.
Thanks. On your plot showing the coherence time, when you tried the XY8, the normalized contrast only seems to go up to like 0.7 rather than 1.
Yes.
Why is that?
That's mostly due to so for XY8 we have amplitude noise. We think we have to average for quite a long time during this interaction. And if the amplitude of the XY8 pulses is changing over time, that'll cause the maximum contrast to decrease. And so there's one of our possible improvements is to, for example, water cool the microwave setup, or to use pulse amplifiers, which better preserve and stabilize the pulse area over time.
Mm-hmm.
Yes.
More questions?
Thanks for this very impressive talk. So just to be sure that I understood this correctly, the light from your tweezer is giving you the dominating quantization axis, so that the ellipticity of the polarization defines then in which direction you have the dipole-dipole interaction?
Yes, that's correct. So actually, this is a subtle point. So for our molecule, there's no n.s interaction. which means that the rotational quantization axis doesn't depend on the B field at all. So even though we're in 860 Gauss, the B field doesn't matter. But there's other molecules like CAF that have n.s, and there, because of that, it's the B field that defines the quantization axis. In those cases, you don't necessarily need to do magic ellipticity. You could do magic angle by having your polarization be linear, and if you've got your p orbital here, it'll be held in place by the B field, and then you can be at a certain angle from it and control the relative E field that goes both orthogonal to the p orbital and along the p orbital. But in our case, because the quantization axis is defined by the tweezer, we can't do something like that. So we have to use the elliptically polarized light.
Okay, good. We have time for one more question, and Mikhail could start setting up.
Presumably, there's some kind of nuclear spin thing going on. Like you were saying, there's no n.s. Is this your 860 Gauss is not enough for the nuclear spins to start being relevant, or is this somehow different?
The rotational state, the eigenstates. There's no n.s, so
there's no like n.I or whatever.
That's also very small. And so the only time that really matters is at particular trap depths. So what you can see here is these crossings where the hyperfine states from other rotational levels intersect with the hyperfine states of these ones. And so these are called avoided crossings. And they actually do matter in a way. So I'll show you this interesting slide here, which is that the transition frequency is affected, the transition frequency is affected by these crossings on the level of 10 kilohertz. And so if you were to plot out like what the optimal ellipticity is for each trap depth or intensity of the tweezer, it varies. And so it's not perfectly true to say that we can be at any intensity. Ideally, we would actually sit somewhere where the slope is quite flat. So yeah, there is definitely this interaction with hyperfine states.
Thanks a lot, and let's thank speaker again.
Thank you.